Okay. Well, thanks again for the invitation, Alain. It's really a great pleasure for me to be here and have the opportunity to discuss with you some of the data we generated in the lab. Do you hear me in the back? Okay, great. So my lab is interested in understanding how mechanical forces are impacting morphogenesis and cell decision in the embryo. And it comes from an observation that you certainly all know, which is the fact that mechanical forces are ubiquitous. They are acting all the time, everywhere, at every scale. So there you go. So they act at every level, every type of species, from bacteria, plants, and mammals. In mammals, obviously, mechanical forces are used for many reasons in terms of sensation. They're also important within our body to modulate physiology. But we are among the people that really believe that mechanical forces within the embryo are also extremely important to modulate shape of our organs. And our idea is mainly to focus on a specific type of forces which are associated with the movement of fluid within the embryo, and address very basic question between addressing the relationship between forces signaling and also shape within the embryo. So in slightly more precise words, we want to understand how mechanical forces regulate gene expression and cell behavior during morphogenesis. And for this, we use zebrafish as an animal model. And I use this slide to just introduce you to different organs which are under the dependence of flow forces for their growth, but also signaling. In particular, early on in the embryo, an axis of asymmetry is generated within a structure which is called the left-right organizer. This is a structure which is transient that appear early on in the embryo, in the tail of the embryo. This is the head. And a flow is generated, and this flow is going to set the asymmetry of organ positioning. And I'm going to talk about this today. But there are many other organs in which flow forces are generated and in which flows will be essential. For example, in the kidney and the neural tubes, as well as in the inner ear, flows are generated. They will modulate morphogenesis. And of course, in the cardiovascular system, where the heart is pumping and generates mechanical forces that will spread in the cardiovascular system and help morphogenesis to happen. Flows are a very good medium to propagate forces, but also signaling molecules. So you sometimes have to find out and find ways to discriminate between the two, which is an issue that is around. Now, obviously, to generate these flows, you need so-called pumps. There are two main pumps in the embryo. The first one is the heart, obviously. And actually, the heart is the first organ to be functional in the body. So this is a zebrafish heart. So the size of a human hair. So very small, but still already extremely efficient in propelling fluids within the cardiovascular system at embryonic stages. And the second structure is an emanation of the cell membrane that we call cilia. It's organized as a microtubule network to create an extension of the cell membrane. And under the right molecular conditions, these extensions are mobile, so there are internal force generated within the cilium, which make them move. And when the movement is well coordinated, you have an helical movement, which is able to generate a flow. Cilia are much smaller, so the scale and the amplitude of the flow they will generate is smaller, but they can work together and their efficiency will add up. So they are also extremely efficient in generating flows in the embryo. So quickly, a small advertising for the use of zebrafish to address cilia function. Zebrafish are transparent, so you have a very high access to live imaging, and it's very important to maintain the animal alive to be able to grab these forces in real time. So these are a couple of examples of movies done in the left-right organizer, in the inner ear, as well as in the vascular system, in which, in that case, it's not a beating cilium, it's just a passive cilium, which will be deflected by red blood cells. We actually think that these cilia are actually sensing the forces at these early stages of vascular development. But overall, a lot of easy access due to transparency of the fish for live imaging, which is great for measuring forces in vivo. So today I wanted to discuss with you about a mechanism of flow sensing mediated by cilia, and in the context of left-right symmetry breaking. So I will quickly introduce you the left-right axis of our body. This is a human representation of our skeleton and our internal organ. And I guess you all know that we look symmetrical, and it's our skeleton which is symmetrical, but within our body cavity, all of the organs are symmetrically positioned, certainly because it helps packing of the organs and put organs into a smaller volume. And this follows an axis, which is the left-right axis, which is set up very early in the embryo. And it's genetically encoded, and the genetic pathways have been heavily studied these last 20 years. So this is an in situ hybridization for genes that are setting the left-right axis in chicken. It turns out that these genes are highly conserved in vertebrates. This is the work that was done in Cliff Tabin's lab. And it shows one transcription factor and one signaling molecule, which are expressed only on the left side of the embryo. This is the lateral plate mesoderm. But you can see here for nodal, the expression is also seen asymmetrically within the structure, which is actually a very small piece of the embryo, which highlights, so the small drawing I made highlight the so-called left-right organizer of the chicken, which suggests that something is happening within the structure that will set the asymmetry of gene expression. It turns out that some decades ago, two Japanese groups, the group of Hiroshi Hamada and the group of Hirokawa, proposed that actually this is an active process which is happening within the left-right organizer. So in mouse, the left-right organizer is called the node. And they could show that this node is ciliated, and the activity of the cilia, it's actually a carpet of cilia which are all beating in a way that they create a flow which is directional and which will go from the right side to the left side of the node. And from these, they proposed different explanation as to how the initial axis of symmetry would be broken. There are actually three mechanisms, and they are still very open, but one of them proposes that it's actually a morphogen. So the flow generated by cilia allows to concentrate a signaling molecule on the left side of the left-right organizer. There is an evolution, if you want, of this morphogen hypothesis, which involves this time sequestration of packages that contain some signaling molecule that will be taken into the flow and get to signal on the left side of the embryo. Alternatively, or in combination, I said that they are convergent, but there is another very interesting hypothesis which involves another type of cilia, which this time are not motile, but more immotile, and they have been proposed to act as sensors, and they have this very interesting localization, which is on the side of the left-right organizer. And one of the hypotheses which was proposed when these cilia were identified were that they were just acting as passive mechanosensors, and they could sense what happened on the left side and trigger the asymmetric expression. So a mechanical hypothesis. So we thought that was really interesting. Just for making you understand how this flow is generated, because this is really the initial symmetry breaking event. It's based on the fact that the cilia are actually asymmetrically positioned on the cell along the anteroposterior side, and through the PCP signaling pathway, this asymmetry of position is mediated. And then their orientation in space is having a tilt, which makes the cilium more efficient in propelling fluids when the cilium is away from the cell membrane than when it's close to the cell membrane. So orienting the cilium in space, making it beat, cilia have to all rotate in the same direction, allows to create this directional flow in the left-right organizer. Now, as I said, this left-right signaling pathway genetically is highly conserved between vertebrates, but also the presence of left-right organizer has been shown to be present in most of the vertebrates, or at least modeled for developmental biology. Interestingly, so they all show signs of coelogenesis, except in one, the chicken, where people have not been able to show that there is a flow which is generated in this structure. I'll come back to that, but so far I have to say that there is not a global way to explain left-right symmetry breaking via cilia motility, but there might be other conservation traces. Interestingly as well, the structure and the shape of the organizer is highly variable or highly different between those species. And we thought that, well, we should check out a bit more carefully what happens in the fish. The reason for this is that it's a cavity which is closed, and we weren't sure how the forces were distributed in this organ. Now, we know from the literature, again, that cilia motility is essential for left-right symmetry breaking. It's true in mouse, but also true as well in fish. If you mess around with cilia motility, you will have left-right defects. And people, through live imaging, again, were able to show that symmetry breaking happens in the cells of the left-right organizer themselves. And the first sign of asymmetry comes from asymmetric activation of calcium signaling on the left side of the left-right organizer. And this is happening within a fairly narrow time period, which corresponds to 2 to 3 hours. So back in the days when I was a postdoc, I thought that, well, this is a very interesting question, but I need to team up with a physicist because they have the truth. And this model is such a physics-oriented system. I need to have some help, and by chance, my neighbor was Willy Supato, which is now at École Polytechnique, and we started to set up some experiments together. The first idea for us was to create a nice approach that would allow us to check out how flow forces are spreading within the left-right organizer. So a few words about the anatomy of the left-right organizer in fish, which is very special. It's located in the tail. Its name is Kupfer vesicle. And this structure is ciliated. So this is a fixed sample where we highlight acetylated tubulin, which marks the cilia. And tough for us, this structure is actually embedded within the mesoderm, between the mesoderm and the yolk, so it's pretty deep within the embryo. So you can see here a Z-scan through the structure, which makes its accessibility not easy, but it allows us to really study the system within its native context without having to explant and put the embryo under the microscope like you would have to do in mouse. So structure, we are zooming in on the left-right organizer here. It's a dye that highlights all the cells of the embryo. It's very nice, spherical structure. It contains beating cilia, as we would expect. Now, looking at the flow within the structure, not that easy because it's transparent, and if you don't have a good contrasting agent, you won't see much happening. Sometimes you will see some particles floating around, but the density of a particle is too low to really have a good sense of how the flow is propagating in this structure. So we set up an approach which was based on femtosecond laser ablation, which allows you to create in a very narrow volume a lesion. You can see here an example of the type of lesion you can get. It's due to the fact that when you generate 2 photons at high power, you can have a diffraction-limited area in which you create a lesion. And our idea was very simple. We could just come, laser ablate one of these cells, create a hole. The cells will be in contact with a dye that labels all the cytoplasm, and we just release the cytoplasm, which is fluorescent. If we are lucky, we'll have some particles that we can track. And of course, we were lucky, and this is the results. So this is a top view of the vesicle. We altered the color coding. The gray is the mesenchymal tissue, and the yellow are the particles which are fluorescent. And the flow is slow enough so that you can really get a volumetric acquisition and keeping the movement of the particles which are seated. Then you can do all sorts of things, tracking, and we could get a fairly nice feel of how the flow is circulating the vesicle. Obviously, it's a circulatory flow, so particles and the flow will go from left to right, from right to left, sorry, but it will come back regularly on the anterior part of the vesicle. Now, the global flow was interested, but we had sufficient resolution to go a bit more closer to what happens at the cell membrane, which is where the signaling would happen. So this is just a summary of how the flow is circulating from right to left. It's actually faster anterior, posterior, and then slower dorsally, slower ventrally, and strong in the middle of the vesicle. But we needed to zoom to see what happens at the cell membrane where signal is happening. And actually, we were very impressed and very lucky. So we could do this, and we could fairly well see trajectories of these particles, where we could track actually those particles and get a feel of what happens in the area where cilia are. Actually, here the cilia are not labeled, but you can see that the particles are circling around in this area, and by negative, if you want, tracking, we could reposition a cilium and see how efficient it is to mix its environment. But it also showed us that, yeah, there is strong mixing, which, in terms of signaling, might not be that great. Now, this is just a volumetric view of the velocity map of those particles. You can see that around the cilium, you have those, we call these chaotic advections, or non-really well-structured streamlines, whereas a bit away from the cilium, you get these nice streamlines, which is basically defining the flow that you saw in the first tracking, which is a nicely ordered flow. But if you go at the cell membrane where signaling happens, this doesn't seem to be very well ordered. It's more chaotic. So this led us to really think about what really is happening in terms of the different hypotheses that were proposed, whether this flow, which is chaotic, is really fitting with the different hypotheses that were proposed in the mouse. So how can we reconcile these models with the flow profile that we see in vivo and in fish? So we simply asked this question: are these flow sensing mechanisms plausible given the flow pattern we observe? And our idea here was to really completely forget about genetics, forget about molecules and gene, and just look at the system from a physical point of view. Just look at the forces and see how they distribute, and then put them back into these models and see whether they are physically alive. So for this, we extended the team. Willie was still, obviously, in the mix. But everything started basically with the arrival of Rita Ferreira, who used to be a PhD student in the lab. And we get more forces on the fluid mechanics side by inviting Andre Wilfen, which is an expert of low Reynolds number fluid mechanics associated with cilia motility, which is exactly the topic or the physical conditions that we are looking at here. So our idea was, again, very simple. Just get all the topological landmarks including the cilia orientation in the vesicle, and then in order to extract the key parameters that dictates the flow forces in the organizer, and then test what could be the potential mechanical mechanism for flow sensing in such a closed structure. So, quick words about the experimental approach. It's all based on live imaging again. This time, we wanted to have a global approach. This is what we needed. So we used a couple of tricks. The first one was to image a transgenic line that expresses ubiquitously a GFP which is coupled with ARL13B, which is a protein that will go within the cilium. And image this line in a way that we could see something that we were looking for, which is an artifact of imaging. Basically, if you scan slow enough, the beating cilium is going to go in the focal volume of your light and go within the focal volume several times. And it's going to create an aliasing effect that will provide us some information about the shape of the cilium and its orientation in 3D. So you can see here a 3D stack of the vesicle. In the middle is the vesicle itself. Around is the mesenchyme. And you can see here those stripes line which highlight cilia which are beaten within the structure. Then, through a little bit of image analysis, we can extract this shadow. So basically, we need to segment the vesicle itself first to make sure that we analyze only the cilia that look within the vesicle. And then using this shadow, we can create a vector from the base to the outside of the cilium itself. And then we just simply collect the 3D information of its orientation. So the next step was a step which allows us to smooth out the data sets in a way that we can compare them from one animal to the other. We found that there was lots of variability of shape of the vesicle itself. So the idea was to use a set of projections of the data sets onto a sphere which would have a single, which would be of a single unit sphere, so we could project all the data sets of individual vesicle onto the same framework. So we could compare then all the data we generate. Now we can do all sorts of things with these data sets by projecting them on a sphere. So this is just a density plot of ornoid projection. Doesn't really matter, but what is important is that through this interpolation, we can now have an access of the variability of the system. Now for display, we used a very conventional approach, which was to take this 3D data and project them in 2D the way you would do it for a world map. And then you can just project the information you want. So here is the cilia density map. We can also project different angles of the cilia. We can combine both orientation plus angles. But I will mainly show you this kind of data. And then finally, we can go back to Andre and provide him with information which are either single embryos or average data from different stages. And he can use this information to run his own simulation, which is an aerodynamic simulation. There is really nothing, let's say, there are absolutely no unknown parameter, if you want. Everything is set. And he can just use the angles and density of cilia to run a simulation within, again, one single sphere, and then measure the flow profile, measure the distribution of shear forces, or if you're interested in, put some vesicles and see how they will travel within that volume, which is modulated by the forces generated by the beating cilia. So then, using this approach, we can test the chemical versus the mechanical sensing, if you want, which defines the three systems described before. So the first thing was to validate the model. So quickly we used the flow profiles, general flow profiles that we generated with the laser ablation, and fitted and checked how the simulation would perform. So the red are the data measurement, and in black it's the flow. It's just so a section of the flow velocity between posterior and anterior. It's a little bit higher here because there are some cilia, so flow velocity increases. And then the center is lower. And then you go anteriorly, and velocity increases. And this was expected because there are more cilia in the anterior part of the vesicle. But overall, we're pretty confident that the model mimics the flow profile which is generated in vivo. And then, so we can go and test one by one the hypothesis. So the first hypothesis we were very excited about was the mechanical flow sensing. So first thing we tested, and we made a number of observations. The first observation, which was disappointing, but on the other hand, we were expecting it, is the fact that, well, flow velocity is similar between left and right. There is no difference between the two sides. The only difference you would have would be the direction of the flow, but in terms of velocities, it's the same. So flow is symmetrical, if you want, except its direction, in terms of velocities at least. Interestingly, we could measure the amount of force which is generated at the cell surface, and it's basically scaling with shear stress. And we found that these forces are extremely low. We are also disappointed, but it wasn't neither a huge surprise. But we could then compare with known mechanosensor, and at least cilia-mediated mechanosensation, and we could see that the flow generated in the vesicle is at least 1 order of magnitude lower than the detection threshold, which is found in developing kidneys. So mechanosensing requires an extremely sensitive mechanism, which to date hasn't been really described, at least in that context. The second observation, which is one of the key, was comparing the flow profile at different stages, but in single embryos. So using individual data sets, now we can run simulations for individual vesicles. And you can see the variability of flow which is generated in this context. And the variability was humongous. You can see here at 3 somites, you have vesicles that have no flow. It's too early. It's too immature. You have vesicles that have high flow here and there, high flow here or here, but nothing there. At 8 somites, maturation happened. The flow is much stronger. But again, the velocity are extremely variable from embryo to embryo. Sometimes you have high velocity on the left, sometimes on the right. So sensing the flow itself doesn't seem to be a very robust approach, at least at these stages, which is the time when left-right symmetry is broken. Importantly also, so the approach allowed us to identify the number and the localization of the immotile cilia, which are the cilia that are supposed to sense the flow forces. And we could see that this number is dynamic. Early on, you have lots of them. Later on, you have much less. Blue are the motile cilia, and red are the immotile cilia. And so, through time, the number of immotile cilia decreases, suggesting that, again, you would need lots of immotile cilia to sense this flow, but you also need high forces to be able to generate enough flow to be sensed. And it turns out that when flow is high, you have lots of motile cilia, but you have much less immotile cilia. So overall, we could conclude that the number of immotile cilia, and we did this through the modeling, was not enough to allow a robust site determination, given the large variability of the flow which is seen in the vesicle. So mechanical sensing, at least from the immotile cilia perspective, looked very difficult, plausible, but very difficult. So we turned to another sensing mechanism, which was never really thought of. But considering the number of immotile cilia is really high, we are wondering what is happening at the level of the motile cilia itself. How are the forces spreading? So for Andre, it was fairly easy. He just model a beating cilium and check the forces which is generated by the effect of this motion. He could measure the torque, so the deformation of the cilium and the flow, and he could say that the force which is generated by the cilium itself is much higher than the force of the global flow which is surrounding him. So by beating, the cilium itself won't be able to sense the global flow. It will sense the flow that it's generating itself. So if it does so, if it senses something, it will sense its own motion and the own flow it generates, which corresponds to this chaotic flow I showed you before. So we weren't sure how this could fit with symmetry breaking per se, but we might have an indication as how this could participate in the system. And we got this idea by just looking at cilia orientation, comparing, so through stages, the left and the right side of the cilia orientation. Again, averaging the cilia orientation and compare left and right. And we could see that even though early on, so this is a map that describes the average cilia angle we could obtain. And these are the plots of that highlights the main orientation of the cilia, looking at their position when they look down. So the vesicle is like that, top down, and we are looking at the angle from that side, from that view, right? And what we see is that early on, still immature, lots of immotile cilia, but still a flow generated. The main orientation of the cilia is symmetrical. Through time, however, what we could find is that eventually the left side kind of deviates from the right side, which remained fairly locked, highlighting an asymmetry that we didn't expect. Interestingly, this asymmetry is resilient to left-right asymmetric defect. It's extremely strongly embedded in the cells themselves. We had much trouble to eradicate this asymmetry looking at some left-right mutants, which suggests to us that it's a chiral influence of the cell itself that sets this asymmetry. But overall, if you think about it, this asymmetry, in terms of cilia orientation, in some complicated ways, but still, there could be some asymmetric information within this asymmetric position that could be sensed by the cells if the cells could sense the cilia on their cilia on motion. However, again, no idea of how it works. Finally, we could test the chemical sensing. I'm looking at Alain because this is certainly why I could come here, but on the other hand, this is the reality. We wanted more mechanical symmetry breaking event, but we weren't very convinced. Now, simulating a chemical secretion in the system was extremely efficient in breaking the symmetry of the organ. This was efficient, so this is the result of Andre's simulation. So you see concentration, color coding. But he had to make two key assumptions. The first assumption is that the signaling molecule has to be secreted in a very narrow area of the system, so that there is a need for an asymmetry in where, along the axis, not the left-right asymmetry, but still, the system has to be patterned in a way that there will be some signaling secretion. In that case, he did an anterior release of the signaling molecule. And the other condition, which is also extremely important, is the fact that once the molecule is secreted, it can signal only once. So once it touches a border, it's going to be active, and then bye-bye, au revoir, you don't want it anymore, because then it would recirculate and signal on the other side. So the stability of the signaling molecule is key in the model as well. So I'm almost ready to leave you with this, but these assumptions actually might be not completely crazy. I just mentioned 2 papers, or actually 3, but a first article which is now on bioRxiv, where people have imaged the localization of vesicles within the cells that make the left-right organizer, and they could find that with time, you can see that the anterior cells of the vesicles contain much more vesicles. Now, okay, we can discuss about this. They have a mutant in which these vesicles are symmetrically positioned, and they have left-right defects. Other observation, this comes from a human genetics. This is actually a work from a group not too far from here, from Imagine, the Thompson's group, who found that a protease called MMP21 is associated with heterotaxia in human, and its expression, in zebrafish at least, is highly concentrated within the left-right organizer. And it seems to be a trend because people have found other proteases which are also exclusively expressed in the left-right organizer that are essential for left-right symmetry breaking. So maybe we have assumptions which kind of make sense, even though those observations are what's worth. Okay, so the conclusions: directional flow sensing is tough. In particular, if you use motile cilia, the shear stress is really low, so you'd have an extremely sensitive system, providing that left-right symmetry breaking is extremely robust. It's difficult, and then you need a very performance mechanosensation mechanism. Now, cells could sense their own cilia motion because we found that cilia are asymmetrical. Still, we see that this asymmetry comes late in the system, so we suspect that it would be downstream of an initial event, even though now we are pretty sure it's a chiral effect. And finally, we are favoring the chemical hypothesis because we think that it's the system that really, in terms of simulation, allows a robust symmetry breaking. So to conclude, these 2 suggestions that were made in mouse are certainly plausible in fish. This assumption, in the way it's described, is certainly not possible, in particular the fact that these cilia should be deflected in the same way as the cilia on the other side. So I'm going to conclude with this. I will just thank the group. We are doing lots of work on cardiovascular development, but of course no time to discuss about it. But this work is mainly the work of Rita in the lab, and we have worked extremely intensively with Willy, Andre, and Frank Juliker as well in that project to make this happen. So thank you very much, and I'll be happy to take questions.